Symposium. Welcome to Leuven. And for those who wouldn't know, oops, wait. So it should work. It's not working. Okay. So for those of you who wouldn't know, so Leuven is hosting the Catholic University of Leuven. And next year, so in 2025, we will be celebrating the 600th anniversary of the university. So this means it's quite old already, as you may have seen from some buildings that you saw yesterday during the walk. But let's come back actually to lobular breast cancer. I think that what is reassuring is that now, over the years, we kind of see a very continuous increase actually in research on that type of cancer. And this is, for example, a search I did 2 days ago on PubMed for searching for lobular breast cancer and lobular breast carcinoma. And you see that really the publications continue to increase, that the number of groups involved also continue to increase. We still need to do better, but that's already a very good sign. I think what we also can say is that finally also we get some increased attention by larger conferences, by other organizations, and I think in the past years, it's very recent, we did have some dedicated sessions on lobular breast cancer in these large conferences, for example, or that we will have. So there is finally also a recognition from the larger community that we need some dedicated attention. So actually, this year I just wanted also to give you, before I go into the practical information, I just wanted also to give you some information about the symposium. So we decided to have a little change in some of the sessions that we have. So we obviously will have fantastic keynote speakers. You will discover them along these 2 days. But then also we decided this year to introduce breakout sessions. And the breakout sessions we will have, you will see this afternoon, 1 block of breakout sessions, and tomorrow we will have also another block. So this is because we believe that while doing this effort to have the only lobular breast cancer symposium together, when we had this opportunity of having all these experts and people interested in doing better for lobular breast cancer, it was also good maybe to take the time to focus on specific questions and specific challenges. And this is why we were making some very specific organizing panels for each of the challenges that we were identifying. And for example, today we will have that on pathological diagnosis, we will have it on imaging, and we will have it on dormancy, 3 topics which we know are very specific and needed for lobular breast cancer. And you need to understand that all these breakout sessions have been prepared already very well in advance by all this organizing committee, which we made sure was always composed by a group of clinicians, scientists, and patient advocates. So it's a new effort, and we welcome everyone then attending these breakout sessions to be also very participative, and we hope that this will generate new insights, new collaborations, and maybe also new works to be done within the field. Then also something that we will introduce this time is that we will have these short pitches, from our poster presenters today, so that people could briefly hear also what all the different posters are about, and they could be triggered also to listen and to talk with these different poster presenters during the poster session this evening. And then finally, we will also have this speed networking event just before lunch, but we will tell you more together with Stephanie Walker later today about that. Don't stress. So now some final practical information. So the Wi-Fi, I think everyone has found. It's also on all the doors and so on. So again, for those of you who require some quiet room, you can go to conference room 6 here at the back of the auditorium. All sessions are recorded. Unless, and will be posted unless the speakers didn't give their consent to. And as yesterday, I would really ask everyone to enter the auditorium a few minutes before the session starts to make sure that we can start on time and that the speakers are not disturbed. Also, I will ask everyone to sign the attendance list every day. We need that every day, please. And then I would ask the speakers and the chairs to make sure to respect the timing of the session, because we have a very full agenda, and I think it's nice to make sure that we can keep the breaks so that people can talk further then and relax. And then finally, at 11, so just before starting the coffee break, we will have a group picture that will take place in the courtyard there. So please, before grabbing your cup of coffee and so on, go there. You will need to look up. We'll take a picture from the window so that we can try to have everyone on the picture. And so now, actually, it is, and also what important is, it's not the last day, but I absolutely also wanted to thank the scientific committee for the help in putting the program together. I wanted to participate, to thank the very active patient advocates for really also putting this symposium together with us, for all the meetings we had, for all the very nice interactions we had also in the development of the many sessions. Then a very, very big warm thank you to the local organizing committee, because without them we could not have had this symposium. And then finally, also, I would like to thank the sponsors, which you see listed on this slide, but also on the banner. So also a very warm thank you. So now I actually would like to introduce the Dynami Foundation. So the Dynami Foundation actually will be supporting the lecture, which will take place in a minute. So the Dynami Foundation is led by Flora Mijianka. I'm not sure I'm pronouncing well. She's a very enthusiastic and passionate person who is dedicating her enthusiasm to try to gather money and attention to lobular breast cancer. And recently, they have developed what they call the Lee Paid Memorial Lectureship. And this year, they have chosen Professor Patrick Dirksen as the person who would benefit from this lectureship grant on the occasion of our annual ILC Symposium. And we are extremely proud of that. So Patrick actually obtained his PhD at the Department of Pathology at the Academic Medical Center Amsterdam in the Netherlands. And following his graduation in 2003, he moved to the Netherlands Cancer Institute to study the consequences of inactivation of cell-cell adhesion in breast cancer development and progression. And there he developed 2 mouse models for human invasive lobular carcinoma, biconditional and mammary-specific knockout of E-cadherin and P53. This is really a very important work which has set the basis for a lot of work on lobular breast cancer. In 2007, he joined the Medical Oncology Department as an independent researcher and head of the UMCU Mouse Cancer Clinic. And in 2010, his position was tenure as associate professor at the Department of Pathology. Today's group is aiming at combining expertise on adhesion molecules, cell signaling, and clinically relevant mouse models to study the downstream effectors of the adherence junction complex that propel breast cancer development and progression. And we know, obviously, that he has a very strong focus on lobular breast cancer. He's also the person who started the European Lobular Breast Cancer Consortium, ELBCC, that Anne Salomon and I proudly are co-chairing. ELBCC aims at better understanding the biological cues that underpin ILC, identify and tackle clinical challenges to improve diagnosis, treatment, and prognosis of this disease. And importantly, since the start, ELBCC is also closely collaborating with many advocates to try to establish a strong EU-based ILC advocacy movement. And finally, Patrick is also a scientific advisory board member of the Lobular Breast Cancer Alliance. So it's therefore my great pleasure to invite Patrick to deliver his lecture here today. Thank you.
Okay, so good morning, everyone. We all know Lee Pate and the magnificent work she's done for lobular breast cancer and, of course, her legacy. So I'm incredibly humbled and flattered that I was asked to do this presentation today. The second European ILC meeting, again in a church. I don't know about you guys, but I feel enlightened. I love to preach, so we're good to go, I think. So today what I want to do is tell you a little bit about lobular breast cancer, how we, or I think, functions from a biological point of view. I will limit myself to the work that is formally experimentally addressed. I will not go into much detail, but if I do, you can forget about them. All you have to do is memorize the concepts that I want to push across and which I think are important for clinical intervention. Okay, so this is the slide I always show at the beginning. It's also because Anne Salomon always tells me morphology is magic and is really important, and you should look at it, and I fully agree with that, but it's also really important to look at the biology. And this is something that was published by Chuck Peru back in the day, defining the molecular subtypes of lobular breast cancer. And why is this important? Because these are the only cues that actually can intervene in a clinical process, so either ER intervention or inhibition by chemotherapy, or recently the immune checkpoint inhibitors. But this is not the full story. These two independent things have to, I think, be combined to have an actual good clinical diagnosis and treatment, and for that I think this is the actual situation. So we have two histological subtypes of cancer, of which lobular is one, a very important one, but also lobular consists of all kinds of subtypes, being the luminal one, the HER2-driven one, and the basal-likes, and that should be taken into effect when designing a treatment. Okay, so these two rascals have defined lobular breast cancer during the war era. So they defined a subtype which they call lobular. I think in hindsight the name is completely erroneous because it has nothing to do with lobules per se. But anyway, they recognized the phenotype and they published this as lobular breast cancer. So this is a bit of a better picture that you all know as well. On the top you see ductal breast cancer, and on the bottom you see lobular breast cancer. And a distinct feature of the difference is that ductal tends to grow in nests, have pushing and invasive margins, whereas lobular have these very particular single file, back in the day called Indian files, type of invasion. And you see, clearly can see single cells. It's a bit difficult to see here if you're not a pathologist, but you can see single cells. However, if you put them into a gel in an experimental condition in the lab, it looks like this, whereas NST gives you an invasive growth pattern that is collective. That is collective, like this. ILC has these single file patterns in single cells. And if you fiddle about with the medium, in this case it's a collagen gel, and you do something with the polymerization temperature of the gel, you can actually induce these typical ILC invasive file patterns. So that's really distinctive for ILC. A bit of history, and I will go back to that in a minute as well. So the first observation that E-cadherin is not present in lobular breast cancer was done by the group of Walter Birchmeier in 1993. You can see that here. This is, oh, I'm pushing the wrong button, sorry for that. You can see here on the top that ILC is E-cadherin negative, whereas this duct is positive. And you can see this is LCIS. The tumor cells are negative, and the cells surrounding it, which are the bioepithelial cells, are positive. So this is the first seminal finding. So the second one was the finding of mutations in this disease. So the first one was reported by Kanai already in 1993, which you can see here. This was only a somatic mutation, so they didn't know at that time whether it was functional. But the real finding came from Geert Berks in the group of Frans van Rooij here in Belgium. They made the actual connection between the existence of somatic mutations and the lobular phenotype. And then in 1979, Carla Vos in the Department of Pathology in Leiden was the first to make the finding that LCIS already contains these inactivating mutations. And this is important because it tells you that already at this stage, E-cadherin is lost, and it's probably causal to the development of the disease. And then 10 years later, during a previous life, In the Jonkers lab at the MKI, I provided formal causal evidence, which Christine already alluded to, by knocking out E-cadherin in a conditional mouse model with p53 in the background, and this induced a beautiful lobular phenotype with all the ring bells and whistles that accompanies lobular breast cancer. So, long story short, it ends up being like this, where ductal breast cancer is usually E-cadherin positive, and lobular breast cancer is usually E-cadherin negative. And we, of course, know this, all of us, and we've developed a white paper last year from the working group, a pathology working group from the ELBCC, where we thought, okay, we have to pinpoint what is the best diagnosis for ILC, because it was still out there with opinionated people using the wrong antibodies. So what we did is use the 5 most prominent antibodies, mapped them, and then found out which ones were the most specific, and out of the 5 we used, this ECH6 and NC38 were the best. And based on this, we provided a standard operating procedure, which we published in Modern Pathology last year, and you can also find it on our website, where we have a very simple designated flowchart on how to differentially diagnose lobular breast cancer from a histopathology point of view. So we want to push this through the channels of the World Health Organization to make this the standard in the field. Okay, so I think it's really, especially within this audience, it's good to have some historical awareness of historic findings. It's just good to know your history, that's one. And I always do this with undergraduates as well, to teach them how to cite proper papers. So if you are telling people about something about E-cadherin or lobular breast cancer, cite the seminal papers from the past and not a review from 2022. At least that's what I propose. So just to put you back into history a little bit, so the first finding that adhesion was actually important in cell-cell contact was done by François Jacob. He's a Nobel Prize winner for something else. But they had a very incidental finding in the lab that I think probably a grad student forgot to put calcium in a medium or something like that. And came back the next morning and saw that these cells were completely dissociated, like this. So this was a finding that back in the day you could publish in Cell, which was really nice, and it's a real seminal finding in the field. And then the next finding was by the group of Walter Birchmeier in Germany. They had an antibody called anti-ARC1, and they mapped it to a protein called uvamorelin, which later turned out to be E-cadherin. If they put the antibody onto cells that were adhering, the cells would dissociate. So that was the first indication that there was something keeping the cells together, and when you meddled with it through an antibody, you would impinge on cell-cell adhesion. Then Masatoshi Takeichi in 1990 cloned the gene and then called it E-cadherin for cell adhesion molecule. I don't know where the IN stands for. And then Rolf Kemmler did the same thing, but he was too late publishing the actual cloning. But what he did do is combine the two. So he combined the calcium finding from François Jacob with the adhesion findings that Birchmeier did, showing that if you mutate the calcium site in this uvamorelin being E-cadherin, the whole system collapses. So this is formal evidence that E-cadherin is really important for cell-cell adhesion. And then the next finding was again by Walter Birchmeier, and they made the first assumption that it has to do something with invasion. So they showed that if you have low levels of E-cadherin, which are D cells, that they are very invasive in a gel. And if you have cells that are very high in E-cadherin, they're not that invasive in a gel. And if you put an oncogene in it like RAS or SARC, then they will lose E-cadherin and start to invade. But this was still an observational observation in that sense, so there wasn't any formal evidence. So the next piece of evidence came from, again, the group of Frans van Rooij in Ghent. I think he's retired now. But anyway, Chris Flemings in his lab made the first actual formal proof that if you do a genetic inhibition of E-cadherin, you get invasion of cells. So what you see here is a very old-fashioned experiment. It's called a chick heart invasion assay, and what you then do, inject cells into the chick heart and see whether they invade. It doesn't invade here, but if you then meddle with E-cadherin expression, you see that it starts invading. So this was the first formal proof of that. Okay, so a bit on what E-cadherin does. E-cadherin keeps cells together. That's what it does in a normal situation and in some types of cancer as well. So what you see here is two cells. In green, you see the cytoskeleton of the cell. In blue, you see the nuclei of the cells. And in yellow, there's E-cadherin. E-cadherin is placed in between the cells. And this is the first point of contact, what you see. And you see the cells start pushing, and that's why you see these wires. So if you do a cartoon close-up of this, you can see that an E-cadherin molecule binds to another E-cadherin molecule on another cell, and then it binds that to the actin cytoskeleton. So it provides the linkage between the cell skeleton and the other cells. That's basically what E-cadherin does. Now you can see, and I told you already that this is calcium dependent. It's dependent on a very specific site in this region. And we modeled this in our lab, and what you see here in real time is the dynamic interactions between one alpha E-cadherin molecule in blue and the other E-cadherin molecule in red. And what you see here in orange are the calcium ions that are needed for the actual interaction for the X dimer. And this is what actually happens in real time. So if you put this down in a cartoon again, it looks like this. And the reason I'm putting an extra cartoon in here is because I have to introduce the catenins. The catenins are molecules that are absolutely essential for the whole function of this complex. First, you have alpha-catenin. You have alpha-catenin that binds E-cadherin to the cytoskeleton, beta-catenin, and delta-catenin, which we also call P120. And I will come back to that in a minute. Take-home message is these catenins are absolutely essential for the stability of the whole complex. Okay, so what happens if a cell encounters another cell and it undergoes an E-cadherin interaction? So the first thing that happens, it touches the other cell with the E-cadherin molecule. And then it starts pulling and pushing. So you get an increase of forces, which then opens up alpha-catenin. It sort of stretches out and then allows vinculin, which is another molecule, to come in. And vinculin is a mechanosensor. So the more tension there is on a complex, the more vinculin will come in. So that's the first thing that happens. And you see that the forces are building up. So the cytoskeleton is pushing and pulling on this molecule. So the next step that happens is that you get recruitment of a factor called ephedrin. Ephedrin binds to alpha-catenin and basically forms a stabilization complex with this alpha-catenin E-cadherin molecule, and then allows vinculin to go off again. So vinculin comes in, tension goes up, vinculin goes out, and the whole system starts relaxing, and then you end up with a stable junction like this. I'm cutting corners here scientifically, but this is the gist of the whole E-cadherin interaction. Now, what about E-cadherin? So E-cadherin is mutated in 60 to 80% of all lobular breast cancers, and we have formal evidence that this causes ILC. So what about the others? So first, P120 catenin, because it's a very prominent member. P120 catenin is absolutely essential for the stability of a classical cadherin. That can be P-cadherin, N-cadherin, OB-cadherin, M-cadherin, all of the classical ones. So we knocked it out together with L. Reynolds. So L. Reynolds did a knockout mouse, we did the breast cancer modeling, and what we saw is that we developed a very high-grade multinuclear metaplastic carcinoma, so we never encountered any ILCs. So P120 is not important for ILC in that sense. To the best of my knowledge, no one has ever tried to knock out beta-catenin. Someone tried it in a polyoma middle T model, but it never developed into breast cancer, which was lobular. And the other thing is that we hardly ever find inactivating or activating mutations in beta-catenin in lobular breast cancer. These two guys are quite important, I think. So it's been now well established that alpha-catenin is very important in this process, not only in lobular breast cancer, so we could show that if you knock out alpha-catenin, you get a lobular-like phenotype, but it's also a germline mutation in hereditary gastric cancer. So it seems that alpha-catenin is one of the key players. We now have the same evidence for ephedrin, a paper that will be published hopefully soon, which I talked about last year. Same thing, if you remove ephedrin from the equation, the whole system is disbalanced and you end up with a lobular type phenotype. And I put a little asterisk there because ILC that is dependent on alpha-catenin and ephedrin ends up to be E-cadherin positive. So these are the 10%, I think, or a part of the 10% that are E-cadherin positive ILCs. And the final one is vinculin. That hasn't been done. Well, it has been done in my lab, but we haven't published it. If you knock out vinculin, you also get lobular-like phenotypes. And we found mutations in vinculin, but they're somatic, and we're not quite sure yet whether they're actually a dominant active or dominant negative. So maybe more on that later in the coming years. Now, all of this is super nice, and we know that this works, but there's a big if and but. So E-cadherin loss is not tolerated in the mammary gland. If you lose E-cadherin, if you model it in a mouse, for instance, all the cells that target E-cadherin are apoptotic. So it's not interchangeable with life. So anybody in the audience know why that is? And maybe the more important question, what should happen before you get cancer? Does anyone have an idea? Scientists can answer the question as well. Ron.
You need a survival factor, a deposit factor, to allow the loss of E-cadherin to be tolerated.
Yes, exactly. So if it's not tolerated, you need something that provides the tolerability. So you need something that basically tells the cell it's okay to lose E-cadherin and you can still survive. So and that boils down to this scheme. Again, it's a bit oversimplified, but I think the take-home message is very valid. First, you need a genetic inactivation or an activation of an oncogenic pathway. In breast cancer, and particularly in lobular, it's activation of PI3 kinase, amplification or mutation of AKT, a deletion of PTEN, or whatnot. I think that's about 60 to 70% of all lobular breast cancers. The other 40 are comprised in part by loss of the P53 activity, either through deletion, mutation, or amplification of MDM2. So if that's the baseline setting, then you're allowed to lose E-cadherin. And that's what happens, I think, in real life. So the main player is E-cadherin. That's 60 to 80%, and then we've been chipping away at these other markers. So catenin alpha-1, so alpha-catenin, ephedrine, vinculin, and the other one is PAR3, for which there is also a little bit of evidence that it induces lobular type invasion. So then you're almost a lobular breast cancer. So we know in the mouse 2 is almost sufficient, maybe a third hit is required, but we know that in humans you need about 5 to 8. So on top of that, you will acquire during the years mutations in other genes, like TBX. I'm a bit of a donkey here, aren't I? So TBX, FOXA, GATA3, and all the others, which are renowned and known tumor genes or tumor suppressor genes, but it is still unclear in what's the effect on the actual cell being lobular and the lobular phenotype. And then, of course, the treatment starts, and treatment is a renowned inducer of additional mutations, such as things we were talking about yesterday, so estrogen receptor 1, I think ERB2/3 is a nice progression factor. Also, we see more mutations in P53 and IGFR1. So it's the first hit, inactivation of the junction, and all the things that basically propel the progression of the disease. Okay, so it's not a one-trick pony. I just want to emphasize that it does a lot of things. So the phenotypes you already know. If you have E-cadherin, usually you get more circumscribed tumors. They are invasive, but locally invasive, at least in the experimental setting. And the reason for that is that if you put them on a plate, they grow like a cobblestone epithelial formation. They stick together. If you put them in a suspension condition, which you see here, most of the cells die. And that's the reason why they don't metastasize in a mouse like this. If you look at the E-cadherin models, it's the other way around. So E-cadherin is lost, you get a very nice lobular phenotype, you get metastases. If you put these cells on very stringent substrates, they are very, very motile, which you see here. And if you put them in suspension, they're okay. So they're really rigid. One cell can survive in suspension for weeks at a time in the lab. You can come back, forget about them, put them back on a plastic plate, and they will grow again. That's typically lobular for you. Now, underlying this is a lot of things. So first of all, we know that mRNA transcriptional profiling is different in E-cadherin negative cells. So that's one thing. We also know that it impinges on protein localization. So an example here is P120. With E-cadherin, it's nicely on the membrane. If you lose E-cadherin, it's translocated to the cytosol and the nucleus, which is a typical ILC thing. We also know that cell cycle progression is affected. So this would be a typical IDC with a lot of phosphorylated RB in the nucleus, whereas lobular breast cancer also sometimes have cells that divide and have phosphorylated RB, but most of the cells have cytosolic RB, although it's phosphorylated, which is really weird. Lastly, something that is hip and trending in the last years is the extracellular matrix. There are clear indications that ductal have a different composition of ECM molecules, which you can see here in green, which are the collagens, the laminins, the fibrillar fibronectins, and so forth. And whereas in ductal, it mostly looks like this, in lobular, you see more of these different patterns. So there's clear differences underlying all these phenotypes. Okay, So diving into the molecular mechanism, and this is important for the things I'm going to talk about in the preclinical setting and in the clinical setting. So first of all, I have to talk about ER. It's not my forte because I don't think ER is directly linked to E-cadherin, and I could fill a complete seminar with ER, but I just want to emphasize that it's the most important thing clinically, right? I mean, the disease is manageable because ER vulnerability is there. There is some specificity between IDC and ILC. I think there's beautiful work from Rina's group, Rina Jeselson and Matt Sikora, showing that there might be specificity for a factor called FOXA1 inducing heterochromatin condensation and super enhancers, which might be specific for ILC. But I'm not going to go into that today. I'm going to stick to what I know, and that is E-cadherin. So what we know is that if E-cadherin is lost, P120 goes to the cytosol. I just briefly showed you that. And the first thing it does, it induces actin contraction. So basically, the cytoskeleton starts pushing and pulling, although there's no upside Q, right? So there's nothing here, but still it starts pushing. And it's very typical for lobular breast cancer cells. We know the molecular mechanism. It's been published a long time ago in 2013. I'm not going to go into that. But the bottom line is that Rho kinase is activated as a central player. So the second thing that happens because of this contraction, you get nuclear localization of a factor called YAP. And YAP is a hippo signaling targeting factor which goes to the nucleus and then basically induces transcription of all kinds of genes. And it's sort of a yin and yang of contraction. If you have high contraction, YAP goes to the nucleus. If YAP is in the nucleus, you will get high contraction. So it's a self-propelling mechanism. And why this is important, I think, is because if you look in normal breast cancer cells, you can see that luminal cells never have nuclear YAP. YAP is in red. Whereas the myoepithelial cells, the ones that are pushing out the milk and have a lot of contraction, they are YAP positive. So a luminal cell shouldn't have YAP in its nucleus, and lobular cells do. So that might be a cue for future interventions as well. We know also that P120 goes to the nucleus where it induces a very broad transcriptional program of differentiation genes that inhibit proliferation. And we published something last year on maybe a rationale why ILC is so low proliferative. So what we found is that a factor called ID2, which is induced upon P120 Kaiser interaction, directly interacts with RB and prevents phosphorylation through CDK4/6. And we think that's the reason why ILC is so slow proliferating in certain settings. Another thing that is really important, but not a lot of work has gone into it already, are the integrins and the ECMs, so the extracellular matrix. So it's not just E-cadherin on the outside, it's also an integrin on the outside. And it basically attaches itself to its environment. So ILC might have specific ECMs, it might have specific integrins, well, it does, but it's not being published that well. And we know that it activates factors like SARC and FAC, which are again upstream of Rho kinase. So this might be a feed-forward loop in the sense that if you lose E-cadherin, things start contracting, you have specific integrins, and once they get to a certain location, they will propel this pro-survival phenotype. And finally, something I want to go into a little bit further today is growth factor receptor signaling. I think growth factor receptor signaling is super important in ILC. Just to show you a brief overview for the people who are not connoisseurs in the group, you have a growth factor receptor that binds to a growth factor. It signals to downstream effectors. One of them is PI3 kinase, which you all know, I think. And then it acts upon a factor called AKT, or used to be called protein kinase B. And this propels, again, phosphorylation and dephosphorylation of all kinds of proteins that are important to the cell. And the bottom line is that it induces activation of either apoptosis if it's inactive or survival if it's active. Now the next slide has nothing to do with lobular per se, but I just want to illustrate how complex and dynamic this is, because you can already see that the feed forward and the feedback loops in here are quite extensive. So this is a couple of movies from my buddy Olivier Pertz in Bern, and what he's been doing is magnificent work on activation of growth factor receptors in real time. So what you see here is a solid tumor, a breast tumor. This is the displacement of the nuclei, so it's just wiggling back and forth. But it's not random, it's completely orchestrated. So one thing you can see here, that the pulses of AKT and the pulses of ERK, which is another downstream target of these growth factor receptors, goes in waves. And it's very dynamic, and it's very orchestrated. And once you put a factor in it like alpeculib, which is a PI3 kinase inhibitor, you can see that the whole thing collapses, but only partially, and the waves are still propagated. And this might be one of the reasons why these drugs are not performing up to standard in the clinic, because there's no real clear benefit in the sense that if you are mutant versus if you are wild type. So these, just to realize that this is super dynamic and complex to look at. It's not on and off and we put a drug and it's gone. It's not like that, okay? So again, this pathway. So what can you then inhibit from a clinical point of view? So the first thing people look at is the proximal thing, and the proximal thing being the growth factor receptor, which is on top. So the first thing that was done was this landmark paper by Chris Lord's group. So magnificent work looking at synthetic lethality, which basically means which factors are important to propel your phenotype in an E-cadherin negative background. So they started knocking out things, and one thing they found was ROS1, which is an orphan receptor tyrosine kinase. And of course, there's a drug against everything, also against ROS. It's not very specific against ROS, but it inhibits ROS, and that is crizotinib. And they found this in the screen as well. So beautiful work showing that there's a vulnerability that can be targeted if cells are E-cadherin negative. And from this, 2 trials were devised. The first one is ROLO, which has been running for quite some time now, using crizotinib at the ICR, and the other one is Rosalind at the Institut Jules Bordet, using entrectinib, which is also a ROS inhibitor. I won't go into the details because A, I don't know them, B, I don't know whether I can share them with you, so go harass either Chris and Philippe about these trials if you want some more information. Okay, so that's one thing. The other important factor is IGF. So both my lab and Adrian Lee's lab have found that if you look at this disease, the one factor that really pops out is IGF. So if you have low E-cadherin, so this is TCGA, but you see the same thing in Metabric and the others, low E-cadherin means high IGF. The receptor levels are about the same, but Adrian showed very nicely that this coincides with activation of the receptor, which you see here in red. So lobular breast cancer has high levels of IGF and higher activity of IGF-1 receptor. So you would think, okay, cool, this is it. I mean, this is clearly very important. There are inhibitors against IGF-1R. Why not use this clinically? Well, there's one big caveat, and that's these things are quite toxic from a metabolic point of view. So if you use antagonizing molecules against IGFR signaling, you meddle with insulin signaling as well, and this messes up your whole metabolic pathway. So this is not the way to go, but I think the promising thing is that IGF is still very potent, and we might want to revisit the monoclonal antibodies against IGF or think out of the box to inhibit IGF signaling in this disease. Okay, so coming back to other things that could be inhibited in lobular breast cancer, I've briefly talked about PI3 kinase. I personally think it's not the way to go. We can talk about this during coffee, but I do think that AKT is very important. And I think it's important because it's very central in this feedback and feed-forward looping activation. So I'm showing you this ridiculous slide because, yeah, it's a ridiculous slide because it shows you everything that AKT activates or is inhibited by AKT or something that inhibits AKT and so forth. The reason I like this slide is because AKT is at the center of the universe, and that's what I wanted to talk about. So, this is a bit about AKT. AKT is a molecule that looks like this. It has a domain that basically does a linkage to the membrane, which is not that important for now. What is important is it has 2 phosphorylation sites. The first one is the threonine, which is activated by PDK1. The other is the threonine, which is absolutely essential for full activation and is activated by mTOR2. And you can already see where the problems start arising, because mTOR2 was identified as a downstream target for AKT, but it's an upstream activator of AKT as well. So it goes back and forth, and that's really challenging. Okay, the good thing is we have a lot of inhibitors. They've been talked about already during this meeting yesterday. The first one I put down here is MK2206, which I think is absolutely the best AKT inhibitor, but it's also super toxic. And that's because it also inhibits mTOR, which is not very good for patients. The ones I'm going to be talking about today briefly are these ones in green, which is, maybe Claire should pronounce these. Where is she? She loves pronouncing these names, no? Okay, so this is called capivasertib, which is AZD5363, and the other one is ipatasertib. And they are pan-AKT inhibitors. So they inhibit AKT1 and 2, which are more metabolic and survival pathways, and AKT3, which is specific for the brain. So I'm going to talk about these as clinical intervention tools. So we know AKT is really important, because AKT was already identified by Chuck Peru as a very central player. My lab and Adrian's lab showed the same thing, but then showed causality. So if you knock out E-cadherin, you see that if you put IGF in the system, you get a massive upregulation of activation. And the experts tell me that you can multiply these signals, so it's about a 30-fold induction. These are mouse cells. We see the same thing in humans. Adrian saw the same thing in humans as well. It's a little bit less because, for instance, this one has a PI3 kinase activating mutation, and then you see a little less activation as if you would if the system were wild type. The inhibitors work really well as well. So in the knockout situation, you have a 10 to 20-fold reduction in increase in sensitivity, so you can use less drugs. And again, this is causal. If you knock it out, you see the same thing. And we did the preclinical mouse experiments in my lab. There's this one in Adrian's lab, and we see the same thing. So they're very effective in inhibiting growth in lobular breast cancer. Okay, so we thought this is really nice. We can do a trial, and I see time is up, but I'm not finished yet. Sabine Lin at the NKI was the lead investigator in this. We wanted to do a trial, AKT and ILC specific using capivasertib. But unfortunately, some of you know the traumatizing experience we had in the group. When this happened, 2 months before the start of the trial, this paper was profit paper was presented at the San Antonio meeting showing the Capitello trial. So basically what they did is exactly the same, but they did it in the whole ER positive, HER2 negative group. And they saw really nice results. So basically, the necessity for Pfizer to do a trial on lobular was completely gone, which is, of course, really saddening. What is more saddening is they either don't read literature, or they don't read my literature, or both. But they had an inclusion criterion that the patients had to be PI3 kinase mutant, or AKT mutant, or P10 mutant, which is exactly the group that will not benefit the most from this treatment, based on the biochemistry we did. The interesting thing is, if you look at the 13-page paper and the 200-page protocol, there's no mention of lobular whatsoever. So I had a talk about this with Mafalda, who's the first author on the paper. She either doesn't want to tell me or she doesn't know, but she told me that there were a surprising number of E-cadherin mutant cases in the responder group. And they will probably publish it somewhere this year or next year. So I'm putting my money on the lobulars. So finally, I'm sorry I'm going over time a bit, but it's just 2 slides. Can we do something with this laziness? So we know there's a very strong dependency on RB phosphorylation in lobular breast cancer, experimentally at least, and in clinical samples. So can we do something for targeting? So there's already some indications that there's a very counterintuitive response to these drugs. So these were 2 papers published in 2009 and 2018, Palbo and abemaciclib, which are 2 CDK4/6 inhibitors. And surprisingly, all the E-cadherin mutant ones are on the left. Apart from SKBR3, which is a HER2 amplified one, the best responders are the ones that proliferate the least, which is, to me, was mind-boggling. So, okay, let's find out if there's an actual causal link between the 2. And we think there is. It's unfortunately still unpublished, but I would like to show with you some data that E-cadherin loss, we think, causes palbociclib vulnerability. So this is a lobular breast cancer that is E-cadherin positive. This is a lobular breast cancer that is E-cadherin negative, and you can see the E-cadherin negative one is way more responsive to the drug. And this is causal evidence. If we knock out E-cadherin in the E-cadherin positive lobular, it gets more responsive to CDK4/6. So we think this is super exciting. And why is this super exciting? Because we can think about combinations. So this is what I think lobular is all about. It's a slow-growing disease. It goes into the bloodstream. It's completely dormant. It doesn't proliferate anymore. It doesn't die. It ends up somewhere where we don't understand what happens. And then somewhere along the line, it starts dividing again. So it's always vulnerable for AKT. It's very vulnerable for CDK4/6. So can we combine the 2 to see whether they are very effective? So this is experimental evidence for this. These are very complicated isobolograms. I think the take-home message here, if you see blue, it's synergizing. And what you see is that the bluishness is most prevalent in the ones that are E-cadherin negative, meaning that palbociclib together with capivasertib is synergistic if you knock out E-cadherin, i.e. lobular breast cancer. And final thing is, we did this also in mice. If you do both drugs, you see this, very nice inhibition. It's not better than ductal, but the fact that it's very effective at all is surprising, I would say. And then we did the actual experiment by looking at relapse, and then we saw a very spectacular thing. So what you see here is the ductal ones. If you wait a while with treatment and then start treating again once they get 300 millimeters, they all progress to rapid disease, whereas the lobular ones remain as a stable disease. It's very toxic, mind you, so we have to still find out what's the best regimen and so forth. Whether you can do this at the same time is also clinically very doubtful, but I think from a proof of concept it looks really promising. So I end with this. ECAT hearing loss is key. It's the thing that's driving the disease, and it's only one of the diseases where we actually know this. So we have to take that into account when developing things. We need a better understanding of the driving mutations along the line. Are they drivers, passengers? Are they invoking chemotherapy resistance, estrogen resistance, and so forth? And of course, an open door is to diagnose and treat according to biology and not just looking at things. So looking at survival pathways, I think, is absolutely key. The use of BH3 mimetics has been completely neglected in the lobular field as well. I think, how is dormancy regulated? What are the survival signals? Is there an extracellular matrix component? What are the immune cells doing in this entire party? I mean, it goes on and on. We need more money, we need more research. And there's the long latency relapses, which is the actual clinical problem. How does that work? Can we target the cell cycle vulnerabilities, chemo resistance and inflammation, and so forth? Okay, final point is that inclusion criteria. This has been bugging me as a biologist for a long time. I mean, if you know that PI3 kinase mutation inhibits your responses, why do you exclude these people from your clinical trials? It's the same with triple negative ILC. Triple negative ILC is not triple negative breast cancer. It's triple negative ILC. So if it has an ILC vulnerability, treat it as such, and not just as a triple negative cancer. At least that's my opinion, but you can take that away however you want. Okay, so final slide. The people in my lab, I want to thank. We have a vacancy for a PhD student, so if you know someone who wants to do a very cool PhD on lobular breast cancer, please come and see me. The collaborators, some of whom are in the room, and the sponsors. And with this, I'm over time. Sorry, Christine, but I'll take your questions. If there is time for questions.
No, we will actually not do questions because but I thank you very much and I congratulate you again for this lecture, which was, I think, really a tour de force and a very nice story actually of everything he has accumulated and a look forward for what we still need to do. So now I would like to invite actually the chairs for the next session. Dr. Philippe Aftimos, Dr. Jason Muabi and Laurie Petiti, so that they could introduce the next speakers. Thank you.
Good morning. As a patient and an advocate, I am very pleased to introduce our first session, which is New Treatment Opportunities. To introduce our guest speaker will be Dr. Jason Muabi. Please hold your questions to the end.
I'm not speaking, but I'm sharing the session. We have two special speakers for this session that we're going to talk about new treatment opportunities. Our first speaker is a legend. He's one of those invisible heroes of mine. He's a professor and deputy head of division and group leader of the Clinical Research UK Gene Function Lab, and he's a professor of cancer genomics at the Institute of Cancer Research in London. He's very passionate about the drug sensitivity and resistance and identification of novel therapeutic targets. Because of his work, I get to open a lot of studies that targets specific nuances in lobular breast cancer. And today he's going to be discussing identification of new targets in ILC. Without further ado, Dr. Chris Lord.
Thank you. So I've got a tough task because I have to live up to that, and also I have to follow Patrick, and as Christine said, that was a tour de force. So thank you. What I'm going to talk about today is some of the principles that we use in terms of identifying new targets in lobular breast cancer. So what I thought I would do, I need to work out how. is just frame the process that we have to go through and establish some of the principles that we try to work to in order to identify new targets in lobular breast cancer, but actually this is true of all subsets of cancer. So obviously we want to focus on targeting areas of unmet need and ideally where these are defined by a predictive biomarker. And it's a no-brainer, secondly, that we need clear selectivity in the targets that we select between tumour and normal tissue. But what we also need to think about, and I think sometimes this is something that is somewhat neglected, we need to think about targets and their ability that when inhibited to deliver sustained responses that are resilient to the constant evolution of tumours and also resilient to the molecular and phenotypic heterogeneity of cancers. And finally, identifying candidate targets can be a relatively straightforward process. Understanding the mechanism by which they work is a bit more complex, but one needs to do that if one needs to identify the pharmacodynamic biomarkers, the patient stratification biomarkers that we need to use if we're going to do informative clinical trials. So in all the things I'm going to tell you about for the rest of the presentation, these are some of the principles we work to. Now, our approach, and I think there are multiple different approaches one could take to target identification, but our approach is to exploit genetic interactions in cancer. So as tumour cells transform from their, to the normal state into their tumorous state, obviously they lose tumour suppressors, they gain the function of oncogenes, and those provide selective advantages to the cell. But at the same time, there's almost a fitness payoff. As Patrick told you, when cells lose E-cadherin, you might think that that gives a cell superpowers and it's hyperproliferative and it runs everywhere and colonizes every tissue. But on its own, that doesn't happen. In fact, it impairs the overall fitness of a cell. So there must be additional cancer-specific adaptations that maintain the fitness of cells in that new state when they've, for example, lost a tumor suppressor like E-cadherin. And if you can identify those cancer-specific adaptations, they can become candidate therapeutic targets. So when we think of these cancer-specific adaptations, we think about these as synthetic lethal or non-oncogene addictions, i.e. those genes where there's a continued reliance upon these cancer-specific adaptations. Now, when we're talking about lobular breast cancer, as Patrick told you everything you need to know about E-cadherin, we're talking in most part about that transition from a normal state where cells express E-cadherin into a state where they do not. Now, there is, when you look in quite reductionist cell line systems, there is molecular rewiring that allows the cells to maintain fitness once they've lost E-cadherin. And just as a kind of almost very simple example, when you take cell lines and inactivate both copies of the E-cadherin gene and you now do mass spec proteomics, you can see a complete loss of E-cadherin, but there's almost a homeostatic upregulation of P-cadherin. And depending on what cell lineage you do this, you see this E to P or E to N cadherin switch. Now in this E-cadherin defective state, these cells are now totally reliant upon P-cadherin. So it just shows you proof of concept that there's a rewiring, in this case of the proteome, probably as a way of trying to maintain some cell-cell contact, and that now becomes a dependency. Now, our other kind of reasoning for targeting E-cadherin is it's an early occurring event in lobular cancers, and that means it's going to be present in most, if not all, clones, and it persists into the metastatic state. It's easy to identify as a biomarker. And finally, it's present in a series of cancers of high unmet need, including lobular. So I'm going to talk a little bit about the dependency that we've been focusing on recently, but use that as a way of highlighting some of the principles I talked about earlier. So using very simplistic isogenic screens where we take multiple different cell line, breast epithelial lines, inactivate both copies of the E-cadherin gene and do drug sensitivity screens, you can see that when cells lose E-cadherin, they become sensitive to crizotinib, the drug that Patrick just told you about, but also to a series of small molecule inhibitors of the Aurora family of kinases. Now, the good thing about these sorts of experiments is it's easy to establish causality. You inactivate E-cadherin and you now see a new drug sensitivity. But what this doesn't tell you is how penetrant or robust that synthetic lethal interaction is in the face of the extensive molecular and phenotypic heterogeneity that you see in cancers. So to get a kind of empirical assessment of how robust a synthetic lethal interaction is, we move into panels of relatively diverse tumor cell lines. So just to show you an example of this, this is work that Rachel Bruff in my lab did, taking over 200 tumor cell lines that have already had drug screens carried out on them, and then classifying them according to their E-cadherin status, which is bimodal, it's either there or not, and then looking for things that predominantly target the E-cadherin effective cell lines. And then you see, again, a series of different Aurora inhibitors. Now, in kind of following this through, this is work that's been carried out by Will Yang, who's got a presentation later and a poster here. Will profiled a series of different Aurora inhibitors, and there are three main paralogs of Aurora, A, B, and C, and found that this Lilly Aurora A inhibitor is probably the most E-cadherin selective of those compounds. And that gives you a synthetic lethality when you take MCF10A cells and inactivate E-cadherin, T47D cells, or even if you look in some of the small number of lobular tumor cell lines that we have. Now that synthetic lethality translates also into looking at the sensitivity, in this case, of leptomeningeal metastatic organoids derived from lobular patients that were derived by Claire Isaac, Andrew Tutton, and Andrew Fitzpatrick, who's here. And also we can see this synthetic lethality when, rather than using inhibitors that inhibit the catalytic activity of Aurora A, if we use an Aurora A PROTAC. So we're somewhat confident that this is not something that's isolated, not an observation that's isolated to just an isogenic cell line, and at the moment we're focusing on now establishing the in vivo synthetic lethality using this Aurora A inhibitor. Mechanistically, as many of you will probably know, Aurora A is involved in centrosome maturation and separation, and it controls the function of the mitotic spindle. And what we see is that when cells lose E-cadherin, there is less Aurora A at the centrosome, and when one now inactivates that residual Aurora A, is probably the cell finds itself in a state that's incompatible with viability. So it's almost like you're leaning on a bruise. And Will describe this more in his talk and his presentation. But when we try to understand mechanistically how is this happening, one kind of relatively unbiased approach we've taken is to do genome-wide CRISPR screens to look for things that drive resistance to the Aurora A inhibitor when a cell has lost E-cadherin. And one of the most profound effects is inactivating a ROGEF, that's also known as ASEF, it's called ARHGEF4 here, that drives resistance. And as kind of Patrick kind of alluded to in his presentation, this is involved in Rho-ROX signaling. And what we think to be our working hypothesis for the mechanistic explanation for this synthetic lethality is when cells have E-cadherin, there's normal P120 and P190 signaling, and that keeps a balance of Rho-GTP to Rho-GTP towards Rho-GDP. However, when cells lose E-cadherin, and as Patrick elegantly explained, there's a dysfunctional, or there's an abnormal P120 function and also P190 Rho-GAP function. That makes that Rho-GDP to GTP balance sit in the side of Rho-GTP. This drives high-level ROX signaling, and that, either directly or indirectly, we don't quite know yet, causes a reduction in centrosomal Aurora A and then Aurora A inhibitor sensitivity. So in doing this and kind of other synthetic lethal work, I just want to kind of walk you through a few things that we've learned, because it's relevant, I think, in terms of thinking whether Aurora A is a good target to take forward, or what other targets we might consider taking forward into either drug discovery or drug development. So the first thing the first principle I want to get across is this idea of synthetic lethal penetrance. So geneticists would use the word penetrance to describe the relationship between a genotype and a phenotype. So for example, how often does a cell with a genotype A display a phenotype B? Or how often does a person with genotype A display a phenotype B? So for example, our genotype could be E-cadherin mutation, and our phenotype could be Aurora A inhibitor sensitivity or AKT inhibitor sensitivity. Now, if you take a panel of cell lines, organoids, PDXs, or patients, in an ideal world, you want the penetrance of your effects to be complete, i.e., and this is a measure of synthetic lethality, if you take your individual cell lines, patients, etc., every time you see the genotype, which in this case is gene A mutant, you want to see the synthetic lethality with gene B. However, what is probably more common is this situation of incomplete penetrance, where sometimes, in some patients, in some PDXs, in some PDOs, in some cell lines, you see that synthetic lethality, but not always. And defining this is quite important when you consider, just to tell you at the moment how how synthetic lethal penetrance is defined. So I showed you earlier, using some isogenic cell lines, you can take out the E-cadherin gene and you can identify some synthetic lethal interactions, but that doesn't tell you how penetrant that effect is. To establish a penetrance, a simple way to do this is take a wide variety of tumor cell lines or organoids or PDXs that have extensive molecular heterogeneity, but you can classify them according to your biomarker, and then ask the question, do those with a mutation have a greater sensitivity en masse compared to those that do not? Now that's quite a lot of work, and in lobular breast cancer, we don't have that very widespread diverse model system, which is an obvious challenge, but it's clearly very important, because even within molecularly stratified populations, there's extensive heterogeneity from patient to patient and also within patients. So we need treatments that are penetrant, that work most of the time the biomarker is present. And also, as I told you before, cancers evolve. So every time every time the tumor population evolves, that's driven by molecular changes. We want to make sure as far as we possibly can that those additional molecular changes don't minimize the synthetic lethal effect. So this is why penetrance is quite critical. And as I kind of told you just now, this is a challenge in lobular breast cancer because we don't have this wide variety of cellular models, organoids, PDXs, GEMs, that we probably have in other subtypes of breast cancer and in other cancer histotypes. So this makes me kind of think, obviously there's a great effort to generate more model systems, but also we could think about maybe moving away from this empirical definition of penetrance to think about predicting penetrance. And I'm going to explain in a short while how we might want to be able to do that. By the way, I just want to point out that this concept of penetrance is relevant when your biomarker is a gene mutation, like E-cadherin, or even when the biomarker that we're using is lobular. You still want to know what the relationship between a patient having a lobular breast cancer and their sensitivity to a drug is before you probably go into trial. So the second kind of concept I want to get across is when we talk about synthetic lethal effects, nearly always we describe them as digenic effects between two genes. In this case, E-cadherin and Aurora A. But the reality is, when you look at this synthetic lethality, and I would argue all of the synthetic lethalities that have been identified in cancer, they are complex, and I'm using that in the genetic term, and polygenic phenotypes. They involve multiple genes. So I told you that our mechanistic understanding of that E-cadherin Aurora A synthetic lethality is actually via P120, P190, Rho-GEFs, Rho, ROC. So you can kind of think about this relationship as not being a digenic phenotype, but a polygenic phenotype, where you have the two genes that are necessary, but in themselves insufficient for the synthetic lethality, E-cadherin and Aurora A, but also the status of all these modifiers is quite important. This isn't just a kind of conceptual thing. This is quite important to work out what your modifiers of your synthetic lethality are, because these are the things that are going to, if they change, reverse the synthetic lethal effect when these things are tested in trials. And so understanding what your modifiers are critical, because they are things that you want to assess as biomarkers in early phase trials. And they also tell you the likelihood of your synthetic lethality being reversed. The third kind of concept I want to get across in this area is thinking about the size of molecular networks. So sorry to get all kind of systems biology on everyone, but if we now kind of think about synthetic lethal interactions on protein-protein interaction maps, I kind of told you earlier that when you think about that E-cadherin Aurora A synthetic lethality, you can see how this probably works on a protein-protein interaction map. And E-cadherin and Aurora A are connected by a relatively small number of intervening proteins, what systems biologists would call nodes. What you tend not to see with a lot of relatively penetrant synthetic lethal interactions are large networks, where your two synthetic lethal partners are connected by 1,000, if not thousands and thousands of intervening protein interaction nodes. And if you kind of think about why is that the case, it's kind of a bit self-evident. The second scenario, where you have a very big, large world network, a change in any one of those components will reverse a synthetic lethality. And that's probably why you just don't see these synthetic lethal interactions involving these large world networks. So thinking about those, your synthetic lethal interaction, is it a small world network? Because the chances are, if it is, it's going to be more penetrant. And just to show you this kind of empirically, some work I did a few years ago with a systems biologist in Dublin called Colm Ryan, we defined the identity of over 200 highly penetrant synthetic lethal interactions using some of the genome-wide CRISPR-Cas9 screens that have been carried out in places like the Broad Institute. So this is nearly 1,000 tumor cell lines. So once you define the list of the highly penetrant, robust synthetic lethal effects, you now start to ask yourself the question, what characteristics do those highly penetrant effects have that are probably less prevalent in those that have low penetrance? And the pervading characteristic is they sit within small world networks. So you can almost draw a graph where you count the number of proteins, interaction partners between your synthetic lethal genes, and that correlates with penetrance. So the fewer nodes there are between your synthetic lethal partners, here shown as A and B, the more penetrant your synthetic lethal effect is. The implication here is you could identify, in principle, synthetic lethal interactions using an isogenic cell line, and then just work out what is the network between the two synthetic lethal partners, count the number of nodes, and predict how penetrant that effect's going to be. So the last kind of concept I want to get across is that we should now get to a point where we're seeking evidence for synthetic lethal interactions in humans before we go into trial, rather than using the demonstration that a drug works as the evidence that that synthetic lethal interaction actually operates in humans. And to kind of explain this, I just want to go back once again to explaining what happens when a cell loses E-cadherin. As I said before, there is an adaptive molecular rewiring that allows cells to survive now that they've lost E-cadherin. And as Patrick told you before, the way he described it was that there's a kind of order of events. Cells, for example, have to upregulate PI3 kinase AKT signaling to allow cells to survive the loss of E-cadherin. Another way of thinking about this is that molecular rewiring that maintains fitness. Those things are synthetic lethal genes, i.e. they're fitness genes for cells that have gain or loss cancer driver genes. And this is a concept known as induced essentiality. They're essentially buffering mechanisms for, in this case, the loss of E-cadherin. So one has to ask oneself, when you look at cancers that have clearly lost E-cadherin, is there evidence that their synthetic lethal genes are upregulated or hyperactive? Because if there is, the chances are that synthetic lethal interaction does operate in humans, and therefore it would make more sense to target that rather than synthetic lethal interaction where there is no evidence in patients. So to try and kind of assess the likelihood of this concept working, with Said Haider, one of my colleagues at the ICR, we carried out a reanalysis of CRISPR screens in over 1,000 tumour cell lines to identify for each tumour suppressor the list of synthetic lethal genes, and in parallel went back through all of the TCGA transcriptomic data and to find those cancers where that tumour suppressor has clearly had a deleterious mutation or been deleted. And then just look for the overlap. Just ask the question, are synthetic lethal genes upregulated, more often than you would expect, in cancers where a specific tumour suppressor gene has been mutated or deleted? i.e. we're looking for these buffering mechanisms. And the answer is yes, and actually surprisingly, or I don't know, maybe not surprisingly, it's not restricted to a particular tumour suppressor gene. It's actually a phenomena you see with nearly all of the tumour suppressor genes, that their synthetic lethal genes are upregulated when that tumour suppressor is lost or mutated. It's also not restricted to an individual cancer histology, but you see it across different histologies, and you don't just see it in the TCGA transcriptomics, you see it across studies. So just to show you as an example, what you can see plotted down here are the list of genes that are synthetically lethal with losing the RB1 tumour suppressor that Patrick just talked about, and those that are hyperexpressed when cancers have lost RB in 5 different cancer histotypes. And you can see that the really profound synthetic lethal interactions are truly hyperexpressed when the cancers have lost RB. And then a completely different example, when you look at cancers that have lost the genes involved in DNA repair by homologous recombination, i.e. BRCA1 and BRCA2, you can see they are also hyperexpressed in cancers where those defects occur. And this is not just true in the TCGA breast cancer data, but it is true in a SCAN-B, a triple negative data set. It's also true in advanced prostate cancer and also high-grade serous ovarian cancer. So it's quite a pervasive phenotype. This also works, and this is relevant here, in the case of e-cadherin. So now when you stratify either breast or gastric cancers according to those that have lost e-cadherin versus those that have not, and then you look to see whether those that have lost e-cadherin, have they upregulated e-cadherin synthetic lethal genes? Again, the answer is yes. So this is on TCGA transcriptomics. Now there's a small number of tumours that have had wholesale proteomics carried out on them from the CPTAC study. So for example, when you look in ER-positive TCGA cancers, and you look at a series of synthetic lethal genes, in general, you see that they are upregulated when in those tumours that have got e-cadherin deleterious mutations. And again, thinking about the Aurora A example I showed you earlier, multiple components in that signalling cascade are upregulated. So I think where we're getting to the point here is we've established that the transcriptomes, at least, and possibly the proteomes of tumours, in part, their composition reflects the tumour suppressor genes that they have lost and the synthetic lethal interactions that are put in place in order for those tumour cells to survive. Again, it's an idea of this adaptive molecular rewiring. So this brings me to my final point. We know that in lobular breast cancer, we do have a relative scarcity of model systems, and that means empirically establishing penetrance by just taking 20 cell lines or 20 PDXs, 20 organoids, or even bigger numbers of those is quite challenging. So maybe rather than this kind of well-worked approach of isogenic screens in a small number of models and then assess penetrance in a large number of models, maybe we should be trying to predict penetrance and the clinical relevance on our synthetic lethal interactions by focusing on effects that have small world networks and focus on effects where there's evidence of buffering in the clinical disease, in the hope that actually what comes out the other end are therapeutic approaches where targeting your target gives a sustained anti-tumor response. So with that, I'll finish. The work that I describe is largely carried out by Fei Fei Song, Rachel Bruff, and Will Yang. And Will's here. He has a poster and a presentation. And I showed you work from Said Haider at ICR and also from Colm Ryan in Dublin. And I'd like to thank our funders as well. And thank you for the organisers for organising such a fantastic meeting and allowing us to present. So thank you.
Excellent presentation. Thank you so much. We're going to keep the discussion session toward the end. Okay. So I'm going to give it to
So now it's my pleasure to introduce Dr. Anbose. So Dr. Anbose is Associate Professor of Oncology in WashU in St. Louis. He had completed his MD PhD at the Memorial Sloan Kettering and then did his oncology training at Johns Hopkins. And since more than 20 years, he is now working at WashU in St. Louis. I learned about HER2 activating mutations in his 2013 cancer discovery paper. And it's my pleasure to introduce him today for a talk called HER2 and HER3 mutations in lobular cancer. Dr. Bose.
Thank you very much. It's an honor to be speaking here to you all today, to be surrounded by these beautiful stained glass. Before I start my talk, just two quick things. The reason I'm wearing a mask is I have a cold, so hopefully you can hear me clearly through the mask. I'm going to be building on some of the topics that Dr. Optimos and Dr. Dirksen have talked about. My talk, when I was preparing this, I primarily aimed it at scientists and clinicians. So my apologies to the advocates. If there's something I say that is not clear, please ask in the discussion section. Okay. Disclosures with Puma Biotechnology and Genentech. So the two largest reasons that HER2 and HER3 mutations are important in lobular breast cancer. Number one is the NCCN guidelines, which are shown here. For those of you who are not familiar with them, the NCCN guidelines are the US guidelines that medical oncologists use to select treatments for cancer patients. And there are guidelines specific to each major cancer type. So there is a breast cancer guideline. There's not one for lobular per se, but there's a breast cancer guideline. In the last 2 years, the HER2 mutations have been included in these guidelines as a target that can be used for patient treatment. Whoops, that's not what I wanted. As you see here. And they say that the presence of these mutations can guide treatment in certain circumstances. We use this in the second line situation after a patient has received a CDK4/6 inhibitor, such as ribociclib, abemaciclib, or palbociclib. The second reason that these mutations are important for lobular breast cancer is because they are enriched in lobular breast cancer. So generally about 5 to 7%, the numbers can be a little higher depending on which study you quote, of ILC contains HER2 somatic mutations. When I say somatic, what that means is it's mutated in the cancer. This is not an inherited mutation. This is not like BRCA1 or BRCA2. When a geneticist looks at the pattern of mutations, what they notice is you have missense mutations where one amino acid is changed to another. Glycine to alanine in the biochemist's notation, for example, at 309. And a pattern of clustered missense mutations is exactly what we see in the well-characterized oncogenes, KRAS or PI3 kinase. So when my group came across this pattern of clustered missense mutations, this was a strong genetic signature that this is an activated oncogene that's driving the cancer. And as Dr. Dirksen alluded to, you have to have a pro-survival factor prior to the E-cadherin loss. And I would speculate, and I don't have any evidence, but I would speculate that in many cases where a HER2 mutation is present in ILC, that this is one of those pro-survival events that precede the E-cadherin loss. So the when you look at all HER2 and HER3 alterations, the percentage of ILC patients affected becomes now 14% when you include HER2 mutations, HER2 amplification, and HER3 mutations. So the outline, the 4 major areas that I'm going to be talking about in this talk. Let's start with looking at the genomic studies, next generation genome sequencing, NGS for short, as well as the preclinical experiments on HER2 mutations. So in 2010, I was working at my university along with Matthew Ellis, our director for breast cancer, on a couple of trials involved with neoadjuvant aromatase inhibitors. So patients were getting letrozole, as you see in the preoperative letrozole trial, POL, or getting a choice of 3 different aromatase inhibitors in the Z1031 trial, which was a cooperative group trial run by the then US cooperative group called ACOSOG. And in these trials, what would happen is a woman with a mass in the breast would be given 4 months of therapy with an oral aromatase inhibitors, and then go for surgery. And the cancer would be biopsied before start of the aromatase inhibitor and subjected to genome sequencing, as you can see here. This was back in the day when we could only do genome sequencing on frozen tissue, before the ability to do genome sequencing on paraffin-embedded tissue, which we now so heavily rely on, was available. This diagram here is a sketch of an individual individual patient's cancer genome. For those of you, again, who like history, it's called a Circos plot, but it's not so much used anymore. But I still find it a very elegant representation of an individual cancer. So we came across a very interesting case as we were conducting this study. Patient number 14 is a postmenopausal woman with ER-positive, gene amplification negative breast cancer. She might probably be termed HER2 low in the modern classification. And her baseline biopsy shows a HER2 somatic mutation at valine 777, which is changed to a leucine. So a simple thing that a molecular biologist will do is pull up the sequence of HER2 and compare it with its close family members. So you can look at human HER2, mouse HER2, human EGFR, which is 85% homologous, and it's the closest related gene to HER2, and you can look at EGFR in the mouse. And what you see is that valine is a conserved valine across those different genes. So that is one clue that maybe that this is an important residue. But when you think about your biochemistry, so if any of you have taken college biochemistry course, they'll run you through all the amino acids and their chemical structures. They'll bore you to death and make you memorize them. But it turns out to be very important. So valine and leucine are very, very closely related. They're both what we call branch chain amino acids, but they vary in length just slightly. There's an additional carbon atom in the leucine as compared with the valine. So leucine is slightly larger than valine. And that has important consequences for the action on the kinase. So when we saw this mutation, we were thinking, well, maybe it does something, maybe it doesn't. Back in 2010, no one had described this mutation before. It was what we now call a variant of uncertain significance. But I looked at this and I said, this could be important. And I convinced my colleagues that we should pursue it. In my lab, we expressed recombinantly the HER2 kinase domain. We assayed it, and I was just flabbergasted when we got this result. You see a 20-fold increase in the activity of HER2 with this subtle mutation. Now, I've been asked, why in the world would such a subtle mutation cause such a large change in the kinase activity? And I like to draw a little analogy. Since we're here in Europe, I'll use an analogy of a Mercedes-Benz. I went through Stuttgart on the way my traveling here. If you want to improve the performance of a Mercedes-Benz, you're not going to lift up the hood and start whacking at it with a hammer. You're going to go in a very selective way, tweak a valve here, make a small change there. And that's how you're going to boost the performance of that kind of an automobile. I kind of view the same way with HER2 and this mutation. So is this an isolated case? No, it's not. In our initial study, we identified 25 patients who had HER2 mutations in their cancer. And what you're seeing here, each circle, each blue circle, represents a patient. And again, as I alluded to, you have the clustered mutations. And I mentioned before the mutation frequency. Higher in lobular breast cancers. We do see them in the 2 to 5% in metastatic breast cancers is all metastatic, independent of lobular versus ductal. Okay, so to quickly run through the science in the lab, so we showed that increased kinase activity, increased signaling in cell lines, and you can now start to define, okay, what is the best drug that we should use for these mutations? We had 2 candidates at the time. This was being done around 2011, 2012. We had neratinib and lapatinib. Both of these, lapatinib was FDA approved, neratinib was in phase 3 clinical trials. We tested both drugs, and you can see that the lower number here for neratinib is very important. It's very potent in inhibiting these mutations. One of these mutations, which represents about a quarter of the mutation cases, is resistant to lapatinib. You can see these bands are still persisting even in the presence of lapatinib. And the cells that are being treated with, that have this mutation, are not killed by 10 micromolar lapatinib, whereas they are being killed with 15 nanomolar of neratinib. So that result suggested to us that neratinib would be the best drug to take forward based on the choices that were available at the time. We did also track trastuzumab, which is Herceptin. I'm not going to show that here for interest of time. So we launched the phase 2 clinical trial, which I called MUTE-HER, for HER2 mutations, to study this. And I'll come back to this slide in a moment. Let me take a segue over into HER3 mutations. So we're working on a very nice collaboration project with Dr. Desmet back in 2015, 2016. Her group was looking at mutations that were more prevalent in ILC than IDC. And they came across the finding of increased prevalence of HER2 and HER3 mutations, also PTEN and AKT, as in ILC compared with IDC. You can see that from the graph here. They're off the diagonal. The mutations in HER2 are what the pattern of what we saw before. Mutations in HER3, and the reason I've colored them so will be obvious in the next slide. They saw this interesting mutation at the end of the kinase domain of HER3. And that was really quite striking. And Dr. Desmet and I were talking at the San Antonio Breast Cancer Symposium, and I said, I know exactly what these mutations are doing. I could demonstrate it. I could diagram it for you. And she invited me to be part of the paper. And so this HER3 mutation here is right at where HER3 and HER2 contact. So if you imagine your hands coming together as HER2 and HER3 contacting, this is one of the knuckles, just right there. So that contact surface mutation was very interesting. And researchers at Vanderbilt University and University of Texas Southwestern went on to show, that this mutation increases the association between HER2 and HER3 and produces growth factor independent growth when introduced into MCF10A cells. Now, the ideal drug to target HER3 mutations has not been defined. There is an antibody drug conjugate called patritumumab deruxtecan that's in clinical trials. Dr. Malabi may be able to talk, or others in the audience may be able to talk a bit more about that. Also, HER3 is going to activate the PI3 kinase pathway. So one could think about alpelisib or capivasertib in the situation of HER3 mutations, but there is no data yet on those. So let's go into what are the data for HER2 mutations? How did this become in the treatment guidelines? So there are 3 clinical trials that have looked at this in using neratinib-based regimens. The MUTE-HER trial was the trial that I and my colleagues launched. So we screened patients, we screened patients for HER2 mutations. If you had a HER2 mutation, you were given neratinib at a standard dose. And like most phase 2 clinical trials, single arm, you continued on it until there was either, for as long as the drug worked, or as long as the patient could tolerate it. The primary objective is clinical benefit rate, as Philippe Aftimos alluded to yesterday. We are including people who have bone-only sites of metastasis, and therefore they cannot be, their disease response cannot be measured by standard RECIST criteria. So by including patients who have stable disease for greater than 6 months, we can include the bone-only patients. This was a 14-institution trial conducted in the US and Canada. Quickly, the patient characteristics. There were 31% were lobular, 94% were hormone receptor positive, estrogen receptor positive, sorry, and that is the pattern of HER2 mutations. It's predominantly seen in ER-positive cancers. All of the patients had to be HER2 non-amplified. That was a trial eligibility requirement because the HER2 amplified patients already have plenty of drugs and options for them. So we were specifically interested in the mutations that are occurring in the non-amplified population. And the patients had quite a few treatments previously for metastatic breast cancer, median number of 3, but the maximum was 10. And we had a nice result in this clinical trial. So one patient, patient number 15 here, her cancer disappeared entirely on serial CT scans. That's called a complete response. You can see the duration of response here. Some of these patients had bone-only disease and stayed on neratinib here for 32 weeks is 8 months. This patient approaching 1.5 years. So the clinical benefit rate was 31%. The median progression-free survival was 16 weeks, and the duration of response was 32 to 47 weeks. And for a single agent, oral drug, in a patient who would have otherwise been offered chemotherapy when this trial was launched, this was an impressive result. Now, granted, we want to go further, but this is proof of concept that neratinib targeting of HER2 mutations in metastatic breast cancer clinically makes sense. So another group at Memorial Sloan Kettering ran a parallel trial called the Summit trial, and gratifyingly, they saw the same results as us in terms of median progression-free survival and clinical benefit rate. Both we and the Summit trial combined neratinib with fulvestrant because the vast majority of these patients are ER positive. And this very elegant paper, which I won't go into for time, shows you kind of improving numbers in the patient responses. And I'll summarize this 2 slides from now. The next step actually was to bring in trastuzumab, Herceptin, because the HER2 could be targeted with a monoclonal antibody. And there's preclinical data that the neratinib trastuzumab combination would be particularly effective. And so this triplet was defined. And that, whoops, well, I guess what you can see here is as you start to build the regimen from neratinib alone, monotherapy, to neratinib plus fulvestrant, to the NFT triplet, neratinib, fulvestrant, and trastuzumab, you're seeing increasing clinical benefit rate, and even more important, increasing median progression-free survival. So patients' cancers are being controlled for a longer period of time with these combinations. And this is the basis for the treatment recommendation. And so I would encourage people to use the neratinib, trastuzumab, fulvestrant combination if possible, if under appropriate circumstances, if they have the HER2 mutation, obviously in consultation with their local physician. And I'd be happy to answer questions about the clinical implications. But the story doesn't stop there. So new directions. So I will talk about an antibody-drug conjugate, trastuzumab deruxtecan, which has been of tremendous interest in the medical oncology community. And at this meeting, Dr. Laura Kennedy from Vanderbilt will be showing her clinical trial on stage 1 through stage 3 lobular breast cancer that has a HER2 mutation. So quickly, the Destiny Pan-Tumor-1 trial that was published about January this year showed that the antibody-drug conjugate, trastuzumab deruxtecan, T-DXd for short, had impressive responses in HER2-mutated breast cancer. I don't know the prevalence of ILC in these particular cases, but you can see a response rate of 50% and a median duration of response of around 11 months. We are now in the process of combining T-DXd with neratinib, and there is previous publications in the field that shows that neratinib enhances the uptake of T-DXd and that it enhances the tumor regression of T-DXd. So what you can see here in a PDX experiment, the vehicle, the control is in blue. T-DXd has a nice effect on its own, but you combine neratinib with T-DXd and you get an even stronger effect that is statistically significant. This trial is a phase 1 safety trial, and so it has what's called a dose escalation. I won't go into that, but I'm happy to answer questions about phase 1 trial design if people are interested in the details. Let's see. My final data slide. So I would like to introduce, give you an introduction on Dr. Kennedy's trial. This is a very exciting trial looking at newly diagnosed stage 1 through stage 3 lobular breast cancer patients. In this trial, they are going to be screening these patients for HER2 mutations. If you have a mutation, there will be a window phase of 4 weeks where you get appropriate endocrine therapy with or without neratinib. So there's a randomization here. There will be a biopsy to determine, does endocrine therapy with neratinib cause greater cell cycle arrest than endocrine therapy alone? Then all patients go on to the combination. So the patients that are on the control arm only get the control arm for 4 weeks and then move on to the experimental arm. After 20 weeks, the patients go for surgery, and they will look at various surgical endpoints of their cancer, the PEPI score, path-CR rate, etc. In conclusion, stage 4 invasive lobular breast cancer with HER2 mutations can be treated with neratinib plus trastuzumab plus fulvestrant. The fulvestrant to be added if they're ER positive. This is what we call a second-line treatment recommendation, so the patient should previously have a CDK4/6 inhibitor. Of course, discuss it with your local physicians if you're interested in this regimen, because they will be able to give you the best guidance. The optimal drug to treat HER3 mutant ILC has not yet been determined. Trastuzumab deruxtecan, either on its own or with neratinib, is being studied for HER2 mutations in breast cancer. And then finally, Dr. Kennedy's trial for stage 1 through 3 patients will be opening up, I believe, in 6 centers in the US, I believe in this coming spring. So I like to, my dog is a very good-natured dog, and he will put up with so much from us. It's just incredible. So I do a lot of events with Susan G. Komen. And this was a fundraiser that we did. Made this humorous photo. And both of us are wearing wigs, obviously. And I dubbed myself Dr. Pink Einstein. And my sidekick is the Pink Poodle. And we thank you for the opportunity to speak here today. And we thank you for your attention. Many, many people contributed to this work. So members of my lab, members of our PDX core, my clinical colleagues without whom all the trials would not have been possible, Dr. Cynthia Ma, Dr. Andrew Davis, Dr. Haesung Park, our phase 1 clinical trial team, and funding. And most of all, thank you to the patients who supported and enrolled in these clinical trials and all of our trials for breast cancer, because we do this for you and we couldn't do this without you.
Questions?
Hi, my name is Jay. I'm from University of Pittsburgh. I also study HER2 mutations lobular. So to my knowledge, HER2 mutation, they can co-occur with HER3 mutation. I wonder what are your thoughts on, or the value of co-targeting HER2 and HER3 for patients with the HER2 and HER2 mutation, and also the feasibility of initiating any clinical trials?
I think that that's a great question. We see a lot of co-occurring mutations in the HER2 mutated patients. And when you think about a cancer genome, even breast cancer, there's probably 50 to 100 somatic mutations that are occurring in most breast cancers. And so HER3 probably adding additional push on the growth factor stimulating, the growth factor axis. I think the challenge, though, is as you're thinking about combining drugs, is how to avoid toxicity. And so that has to be very clearly mapped out. We have not looked at the HER2, HER3 co-mutated combination per se.
Thank you.
Steffi Österreich, also University of Pittsburgh. Fabulous session. Thank you so much. Chris, 2 questions for you. One is on the upregulation of the synthetic lethal dependencies. How do you know that's a true dependency, whether that is something ILC lineage specific, or do you think that's the same? And the second question I have is more related to all these different nodes, right? You have so many nodes, it's very hard to predict resistance. Do you think it's feasible at all, instead of sequencing and predicting, that you can have a functional assay to do that in a short term, let's say, read out in an organoid or something?
So to answer your first question, yeah, if you look in the transcriptomics of any tumor, many of the things that are upregulated, downregulated, are probably coincidental to the fitness of the tumor. I mean, we're defining those things as being dependencies purely based on the almost the preclinical functional work. Now, whether some of those things are upregulated because that's a lineage thing, and it's somewhat difficult to do separate the fact that in breast cancer, most of your E-cadherin mutant cases are lobular. But to be honest, I think the way we're using that concept is not to say necessarily one should target something, and it's likely to be reflected as a homeostatic mechanism, but actually to focus on things we shouldn't probably go with. So I don't think one can separate those two things of lineage versus the E-coherent loss, because in any patient cohort you've got lots of co-segregating variables. Sorry, you have to remind me what the second question was again.
Second question is prediction of resistance functional assays rather than...
Yeah. I suppose it depends what the mechanism is. I mean, I think probably another way potentially of not so much assessing how resistance might emerge, but maybe a more empirical way of assessing the likelihood. If one does, for example, CRISPR screens, I mean, this is a preclinical approach, as well as looking at the things that when you inactivate them or activate them drive resistance, you just count the number of ways. You know, how many different proteins can I modulate that give me a resistant phenotype? And if the number starts to become very, very high, statistically, the likelihood that the cell will adapt and one of those mechanisms will emerge, I think it's probably quite likely. But in terms of a functional assay that one could use in ex vivo to predict resistance, I'm not sure we're quite there yet.
Sean Egan in Toronto. I was interested in the HER3 mutations, and I know you may not have a lot of them, but do those tend to be found in primaries but not METs, whereas HER2 mutations are found in METs, just because HER2 can activate PI3 kinase signaling through HER3, but also activate RAS pathway on its own, so RAS being associated with metastasis. Is there a difference in survival or presentation?
Yeah. So I have not worked specifically on the HER3 mutations. I've really focused on the HER2. We think some of these cases are truncal, that they are present in the primary, though we have not defined the exact percentage. There are known to be acquired HER2 mutations as well, and the work of Nick Woggle at Harvard MIT has looked at that. I believe it was a 2018 or 2019 paper in Nature Genetics that looked at acquired HER2 mutations, so as the cancer is undergoing treatment. But we think that at least that a sizable percentage of these are present in the primary.
Maria Karsten from Charité. Just a question regarding resistance. With the arrival of ribociclib in clinic in the adjuvant setting, I just would be interested in your clinical opinion, because I feel very, reluctant, or I'm conscious about giving patients who would, with an ILC, giving ribociclib in the adjuvant setting in light of the PADA-1 trial and resistance mechanism. What is your recommendation regarding that?
So let me take a first crack at that, and then I will pass the microphone down, because Dr. Moabi or Dr. Aftamoso may also have some comments. So I'd say my philosophy, my inclination is when resistance is developing, I'd like to know why. What are these particular alterations that are occurring? Is it a loss of RB1? Is it an activation of PI3 kinase AKT? Is it an activation of HER2? Because knowing what is triggering the resistance to ribociclib will then dictate your next step. So I would speculate, and I would, that if there was found to be if there was found to be a HER2 mutation or PI3 kinase AKT1 mutation, that one try to bring in a drug specific to those mutations as the second line. Now, if it's an RB1 loss, that's a slightly different situation. Let me pass the microphone down and to the others. Did you want to say anything?
I'm not a clinician, so
I would say it's different when you talk about the early setting than the metastatic setting, right? In the metastatic setting, you have the cancer that's very active, and the cancer is constantly trying to find way to bypass what you're trying to do to it. In the adjuvant setting, this cancer is not very active, right? Because hopefully we would have staged those patients, make sure there is no active metastatic cancer. So here you're more, if there are what we call seeds, right? Those are the ones that try to make problem in the future. It's to keep them in check, and those will not usually develop those type of acquired resistance. And case in point, a lot of time when we treat with AIs and the cancer came back, you don't have ESR1 mutation right away. In the vast majority of cases, it's acquired as a treatment selection once you start treating metastatic cancer. So I wouldn't be worried about acquiring those mutations in the adjuvant setting. I would be worried about them in the metastatic setting.
Just to add to that, from PADA-1, I mean, those were patients that were developing ESR1 clones on an aromatase inhibitor and CDK4/6. But we also have data from other studies that show that patients with ESR1 mutations can still respond to an AI and CDK4/6. We have also currently studies with novel SERDs in the adjuvant setting, either upfront or switch. So definitely, I think in the future, the landscape is going to change, but they will not replace adjuvant aromatase inhibitors, as some of these adjuvant studies are allowing patients to stay on our adjuvant CDK4/6 inhibitors.
So you would give the ILC patients also the CDK4/6 inhibitors in the adjuvant setting?
I would, based on their stage. I mean, for Abema, it's a higher stage. For Ribo, it's stage 2, stage 3. In Belgium, we don't have access to adjuvant Ribo. We have access to adjuvant Abema. So I don't differentiate between lobular and NST when I give adjuvant Abema.
Okay. Thank you.
Hello, and thank you very much for fabulous talks. They're very inspiring. One of the things that concerns me as a patient is that 40% of us, at least, I think, get ESR1 mutations. And you're beginning to talk about new drug combinations, which are absolutely brilliant, but they always seem to be tied to fulvestrant. And if fulvestrant is effective, then why is there a race to get out lasofoxafen, elacestrant, camizestrant, and in toxicity reasons, camizestrant's already been tested at 150 and 75 milligrams, so less toxicity, it's just as effective at both levels. And you've got imlunestrant. I mean, there's lots of zestrants. And it just concerns me why we're tying everything to fulvestrant, and when are we likely to see a change? Because it'll be very interesting to see those wonderful results. I looked at your monotherapy, Professor Bowes, and I looked at your one with fulvestrant as well, and both results were good. And I thought, well, how would that change if you were able to swap out fulvestrant for a new, different, even maybe personalized? Because, for example, lasofoxafen has an exquisite sensitivity to Y537S. And so if your ESR1 patients could benefit from that, and generally in trials going forward, is this something we can ask for? I know there's lots of trials already with these drugs, but I just see a lot more coming out with fulvestrant, and it's something that concerns me. So if you could put my fears at rest, that would be brilliant. Thank you.
So, wonderful question. So let me give you a view into two aspects of clinical trial design, or clinical trial or drug approval. So probably around 2015, I was sitting in a meeting, myself, the director of our breast cancer program, and the CEO of the company that had neratinib. And we knew we needed a drug combination, and we were batting around ideas as to what to combine it with. Now, this is 2015, so the novel SERDs were not available yet. And the CEO of the drug companies was saying, you know, the ideal candidate to combine neratinib with would be a drug that's already FDA approved for metastatic breast cancer, because it is a simpler process to add to an approved drug. Further, the approved drug would be paid for by insurance and would not have to be paid by the company as part of the conduct of the clinical trial. So it reduced his cost. That was his rationale for how we looked at clinical drugs. I was feeling at the time that we should have picked trastuzumab, but trastuzumab was not FDA approved in that particular situation, and it would have required that the company pay for the trastuzumab for everyone on the trial, something that they were hoping to avoid. So that's one consideration in how these decisions are made. Now, granted, this is 2015, before all these new drugs came about. So now as the companies are making these new drugs, one could certainly go to them and say, I want to see new combinations being tested. But you have to also realize the next thing that the drug companies are thinking about after cost of the trial is regulatory approval. What is the regulatory body, the FDA in the US or the EMA here, what is their bar going to be? How are they going to judge this clinical trial? Are they going to give it regulatory approval? And in the regulatory approval, they want to know two things. Is the combination safe? And is the combination effective above, the individual drugs themselves? And so that are going to be things that drive the trial design and drive the decision making. Now, one could say, why do we have to do all this? We know that drug A works for this mutation, drug B works for that mutation. Why don't we just put them together and give it to the patient? And I would say, please, please, please don't do that. The reason is, when you start making untested combinations, putting them together for an individual patient, you risk toxicities that are unpredictable. You don't know what you're doing. And there was a professor in North Dakota, somewhere in the western US, very senior guy, who was doing this with the help of a pharmacist. It was quite questionable. And we saw this, he was talking about it at ASCO, and I was chatting with Dr. Eric Weiner, the former ASCO president, the director of the Yale Cancer Center now, was there. He overheard this conversation. After the person stepped away, he said, that's very irresponsible. So we cannot just pick drugs off the shelf, combine them together. It has to be done in a well-controlled clinical trial. It has to meet the requirements of the appropriate regulatory body.
Thank you. I think that's all the time we have, but our guest speakers will be around, so please feel free to grab them at lunch. Thank you again to everyone on this session.
So thanks again, everyone, for this beautiful morning session. Just some